Hello, everyone, and welcome to today's webinar titled Armoring T Cells with Engineered Fusion Proteins Enhances Therapeutic Efficacy Against Hematological and Solid Tumors. This webinar is a part of the 10th Annual Cancer Research and Immuno-Oncology Week. I am Antonina Salcido of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Stem Cell Technologies. For more information about stem cell technologies, please go to stemcell.com. Now let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract window on the sidebar to the left of the presentation and follow the process to obtain your credits. I now present today's speaker, Dr. Shannon Oda, Assistant Professor at the Bentown Center for Childhood Cancer Research and the University of Washington School of Medicine. For a complete biography on our speakers, please visit the Biography tab at the left of your screen. Dr. Oda, you may now begin your presentation.
I would like to thank the organizers for the opportunity to talk about our work today, where we will be discussing armoring T cells with engineered fusion proteins to enhance therapeutic efficacy against hematological and solid tumors. Here are my disclosures. So it's an exciting time for cancer immunotherapy, where several basic immunological discoveries have led to breakthroughs in the clinic, which has led to an increasing appreciation that the immune system can be harnessed to control and eradicate cancer. And this has been shown in multiple publications now, and also in the Nobel Prize awarded in 2018 for the discovery of immunotherapy by inhibition of negative immune regulation. So in my lab, we work to restore the anti-cancer immune response with T-cell adoptive immunotherapy, which we like to call a living drug. And so what I mean by that is that we isolate blood from the patient and we get T cells. We remove those T cells from the blood. We genetically engineer those T cells in the lab to target them to tumor. And then we grow millions of those T cells that we can then infuse back into the patient so that those living cells can migrate throughout the body to search out and destroy cancer. So there are two major types of adoptive cell therapy T cells. The first is chimeric antigen receptor, or CAR T cells, and these are fairly well known for their success against CD19 positive tumors. And with CAR T cells, you can target surface proteins, which represent about 27% of the total proteome. These are HLA non-restricted, so you do not have to match the antigen receptor with the HLA type of the patient. We commonly use TCRs instead, or T-cell receptors. So with TCR T cells, you're able to target both intracellular and extracellular peptide antigens. These are HLA restricted, so you do have to match the HLA of the TCR with the patient's HLA, but there are several antigens that are being targeted by this method, including NY-ESO, WT1, and mesothelin. So in a recent paper from Eric Tran's group, he showed that TCR T cells can be effective against solid tumors. So here we're looking at images of a tumor before treatment and after treatment with TCR T cells, showing significant regression of this pancreatic tumor nodule with the TCR T cell therapy. So with the TCR T cell platform, even with finding a great TCR or a CAR, we know that there are still several obstacles in the tumor microenvironment that can inhibit the anti-tumor T cell response. So T cells can compete with tumor cells for key nutrients. There's often a lack of costimulatory ligands in the tumor microenvironment, which could provide a positive signal for T cells. Instead, there's often inhibitory ligand expression and soluble factors that inhibit T cells. The tumors can downregulate their target antigen or HLA, and T cells themselves can become exhausted And this can be further potentiated by the lack of missing or the lack of supportive cells in the tumor microenvironment. So in my lab, we asked how we can armor our T cells to overcome these obstacles. And rather than address each of these obstacles individually, we're looking for opportunities to develop engineering strategies to address as many of these obstacles as possible within one carefully engineered approach. So we develop novel engineering strategies using engineered fusion proteins, which combine two native proteins to form a new hybrid protein. So we take the extracellular domain of one protein and combine that with the intracellular domain of a second protein to form a new fusion protein that is not found in nature, but is formed from two native proteins, resulting in a very low immunogenicity approach. And with this approach, we can deliver co-stimulatory signals, which can initiate gene expression programs in T cells to counter several obstacles. So co-stimulatory signals can enhance the proliferation of T cells or their ability to multiply in the patient. We can get enhanced tumor cell lysis, resistance to exhaustion, increased longevity and memory formation, as well as altered metabolism of our T cells, dependent on the co-stimulatory signal that we select. So as I mentioned, one of the issues within the tumor microenvironment is that tumor cells can express proteins on the surface of the cell that shut down the T cell. So when this inhibitory protein is expressed by the tumor cell or other inhibitory cells within the tumor microenvironment, it can bind to the inhibitory receptor on the T cell and shut down the T cell, and impeding it from lysing tumor cell. So one of our approaches is to essentially replace a brake with an accelerator, where we are rewiring inhibitory or death receptor signaling. So to do that, we take the ectodomain of an inhibitory or death receptor, and then we replace the intracellular domain with a co-stimulatory receptor signaling domain. So now instead of an inhibitory signals, T cells get a stimulatory boost from the switch receptor. So when the T cell binds to this inhibitory protein, the switch receptor delivers a positive signal. The T cell is able to again lyse tumor. So we have now shown that switch receptors can enhance multiple T cell functions, including T cell proliferation. So here we're showing with a FAST1BB switch receptor that we published that we can get robust proliferation of our T cells relative to TCR-only T cells. And this is to a very low level of stimulation. We can get metabolic reprogramming. So here we're showing increased mitochondrial biogenesis with a FAST1BB engineered T cell. And we can also get increased production of functional cytokines from our human T cells. So this is human T cell data showing that we're able to get robust quantities of TNF alpha and IL-2 production from our switch receptor engineered T cells. So one of the cancers that we focus on in my lab is acute myeloid leukemia, or AML. And this is an important cancer to develop new strategies for, as the survival rate lags behind other blood cancers. So the 5-year survival rate is around 26% for AML patients. We also work on pancreatic cancer in my lab, and this is also a very serious disease that has a survival rate that has not improved. So in this chart, you can see the changes in survival from the 70s to 2010, 2011 for a variety of cancers. And this reflects improvements in early detection as well as therapeutic options. But pancreatic cancer is down here at the very bottom with about a 10% 5-year survival rate. And it's predicted to become the second most deadliest cancer by 2030. Some of the challenges with pancreatic cancer are that early detection is uncommon. Pancreatic cancer tends to spread quickly and recurrence is likely. So one of the targets that we wanted to go after with our switch receptor platform was CD47. And this is a very highly sought after target as it is overexpressed in most adult tumors. So here we're looking at overexpression of CD47 in the tumor tissues shown here in the red bars versus normal healthy tissue in the black bars. CD47 is also overexpressed in pediatric tumors. So here we're showing CD47 expression by mRNA in a variety of pediatric tumors. Specifically in pancreatic cancer, CD47 is significantly overexpressed, and high expression correlates with reduced survival. So in panel B, you can see that the patients that have high CD47 expression in red have a worse survival than the patients that have low CD47 expression in the blue line. So the SERP alpha CD47 interaction can inhibit antitumor immunity by multiple mechanisms. One mechanism is that this axis inhibits macrophages from phagocytosing cancer cells. And another mechanism is that CD47 signaling can inhibit dendritic cell antigen presentation to T cells. So there are multiple opportunities for us to address this axis to improve our anti-tumor immunity. So CD47 has been targeted by antibodies, and shown here in this mouse model, these antibodies modestly controlled pancreatic tumor progression. However, one of the issues with CD47 targeted drug therapies is that they can cause anemia issues, as CD47 is also expressed on red blood cells. And so if you block that signal, then macrophages can phagocytose normal, healthy red blood cells. So our approach is to develop a SERP alpha CD28 switch receptor that combines the SERP alpha ectodomain with CD28, which is a co-stimulatory signal for T cells, to develop this new fusion protein to address the CD47 overexpression on tumor cells. So to develop this switch receptor, I developed a panel of fusions that combined different sections of SERP alpha with CD28. So shown here in the red are the sections of SERP alpha that were incorporated, the extracellular domain, and with the first fusion also extending into the transmembrane domain. And then in the blue are the portions of CD28 that were included in these different switch receptors. So the last four switch receptors have CD28 that extends out into the extracellular space and includes an extracellular cysteine. And this is to promote dimerization of the protein or the receptor, which is known to enhance CD28 signaling. Additionally, what we've shown in our previous work is that it's important to also consider the size of the ectodomain of the switch receptor. So the space between the T cell and the tumor cell is actually a very narrow space where the immunological synapse is. And so in order for our switch receptors to co-localize to the immunological synapse, which is where native CD28 signaling occurs, we want to have appropriately sized ectodomains. And so what we've shown previously is that if you have too large of an ectodomain, it's effectively excluded from the immunological synapse. So with these switch receptors, we also took that into consideration. And so we truncated the ectodomain of SERP alpha to accommodate those extra amino acids from CD28 in the last two fusions on the right. So the first, second, and then the red fusion preserve the T cell tumor cell synapse distance. So similar to what we had previously discovered, we found that SERP alpha CD28 constructs that are engineered to maintain the T cell tumor cell distance exhibited enhanced proliferation. So those three fusions that I showed on the previous slide that are appropriately sized to fit with an immunological synapse were able to promote proliferation of our engineered T cells in the red relative to our TCR only controls in the blue. We also did multiple stimulation assays, which we found to be effective in determining which switch receptors are developing or promoting the best costimulatory signals. And with this assay, we found that there were two fusions in particular that exhibited increased ability to accumulate T cells upon multiple stimulations. So both the SERP alpha transmembrane fusion and also the SERP alpha fusion with the -9 amino acids that had the truncated SERP alpha to accommodate those extra amino acids from CD28. So we tested this in a mouse model for AML, and we found that whereas the switch receptor that had the SERP alpha transmembrane did not greatly improve therapeutic efficacy relative to our TCRT only in the black, our SERP alpha -9 amino acid fusion did significantly enhance therapy relative to our TCRT only. So we continued with these studies with our pancreatic cancer cells. And so this is an assay looking at the ability of our TCR-T cells or our switch receptor TCR-T cells to lyse pancreatic cancer cells in an IncuCyte assay. And so we found that the leading fusion had provided much better ability for our T cells to lyse tumor relative to our TCR T cell control in the black. So to test this strategy in vivo with solid tumors, we used the KPC genetically engineered mouse model of pancreatic ductal adenocarcinoma. And this mouse is a fantastic model. It has mutations in KRAS and P53, and this recapitulates 80% of human pancreatic cancers, which also have mutations in these two genes. So these mutations are held under a pancreas-specific CRE. So these mice spontaneously and continuously generate tumors from epithelial cells in the pancreas. So because these mice are constantly developing new tumors, we'll never be able to cure these mice, but hopefully we can shift the curve with our approaches in this model. The KPC model has been used to screen drugs to predict therapeutic efficacy in patients. So that's very important for us to be able to determine what's effective in this mouse model and be able to move it into clinical trials. And the KPC model recapitulates several barriers to T cell function of human PDA, including a suppressive microenvironment. So it has been shown that mesothelin is a tumor antigen that's reliably overexpressed in the various stages of pancreatic ductal adenocarcinoma, as well as in the KPC mouse. However, with mesothelin targeted T cells, even with that targeting, it was shown in this paper that those T cells become rapidly dysfunctional in the tumor microenvironment. So by day 8 post-transfer, these T cells are already less able to make effector cytokines, including interferon gamma and TNF alpha, relative to their counterparts in the spleen. So to use this model, we screen the mice by ultrasound to determine when tumors arise. So on the left, we have a healthy mouse with a normal pancreas, and on the right side, you can see that there are tumor nodules, and so we enroll those mice to study. So once those tumors are detected, we enroll the mice to study as we would do patients, and then we give them an enrollment dose of a single dose of cyclophosphamide, which is similar to what we do in the clinic, and then we give them a dose of T cells, as well as mesothelin as an antigen boost. Day 7 post-transfer, we assess the T cells in the blood and we check their tumor burden by ultrasound. And then we give them an additional 2 doses of T cells without cyclophosphamide for a total of 3 doses of T cells. And then we check the mice for the duration of the study. So importantly, this is a very challenging scenario for our T cells. We're not giving them IL-2 injections. We're not giving the mice IL-2 injections in vivo. So we're not supporting the T cells with IL-2 in vivo. They must be IL-2 independent, or at least from exogenous IL-2. And we're limiting the number of T cell transfers to 3, whereas previous publications that have used this method have done T cell injections every other week for the entire study. So we first checked for safety. So we looked for erythrocyte concentration within the blood to determine if CD47 targeting would have the same off-target issues as with the CD47 targeted drugs. But we do not see any erythrocyte depletion, supporting that our approach is not resulting in off-tumor targeting of red blood cells. We did some histology with our tumors and found that our SIRP-alpha CD28 TCR T cells have reduced tumor stroma, as shown in the reduced area of green in the far right panel, and increased levels of healthy tissue in the yellow. And that is relative to both the TCR T cell treated mice in the middle, as well as the no T cell treated on the left. But what was really exciting was to do the survival study and see what a significant shift of the curve that we achieved with our SIRP-alpha CD28 TCR T cells. So we were able to increase our median survival from 31.5 days to 77.5 days. And you can see the significant shift of the red curve relative to the TCR T cell control curve in black. So very excited to see these results and continue to develop this publication to submit later this year. So some conclusions from the switch receptors are that CD47 is overexpressed in most tumors, and increased expression of CD47 correlates with reduced survival in pancreatic ductal adenocarcinoma. Switch receptors can rewire inhibitory signaling from tumor cells to convert a negative signal to a positive one. And CD47 targeted switch receptors can enhance T cell therapeutic efficacy without toxicity. So we developed a powerful engineered protein platform, the switch receptors, to provide costimulatory signals, but only to engineered T cells to significantly enhance anti-cancer function. However, we know there are multiple types of immune cells in the body that can be directed to eradicate cancer. So we asked if we could next generate a new platform to boost both T cells and catalyze an anti-cancer response from other immune cells in the body. So our new platform we've called dual costimulatory receptors, or DCRs, and these combine a costimulatory ligand ectodomain with a costimulatory signaling endodomain. So by including a costimulatory ligand ectodomain, we can provide co-stimulation via costimulatory receptors on the T cell or on other cells. And we still have the costimulatory signaling endodomain, where we can provide signals that may not be safe to deliver systemically and should be restricted to those T cells that we've targeted to the tumor with an antigen receptor. So with this approach, we asked if our DCR T cells could stimulate a diverse anti-tumor response by providing costimulatory signals to dendritic cells, endogenous T cells, B cells, NK cells, and so on. So first we asked if DCRs can enhance T cell function in a cell intrinsic manner. So we engineered human T cells with our DCRs and stimulated them in culture. And what we found is when we looked at CD9 expression, which is an activation marker for T cells, our DCRs did indeed promote increased activation in human T cells. And then that is relative to the full-length costimulatory ligand, where we just engineer the full-length ligand without any modifications. By adding in that intracellular costimulatory domain, we see enhanced activation of our T cells. We also tested our DCRs with human pancreatic cancer cells. And so whereas the unmodified T cells as our control do not impact tumor cell lysis, we found that our full-length unmodified control also did not promote these T cells from lysing tumor in this particular assay. But really, the effective DCRs are much more capable of lysing tumor, and that is shown here with the ability of these T cells to lyse the tumors, shown here by the reduction in tumor signal. So next, we asked if DCRs can enhance the function of other immune cells in a cell extrinsic manner. So to do that, we generated immature dendritic cells from THP1 cells, and then we co-cultured those immature dendritic cells with our DCR T cells to see if we could get enhanced dendritic cell maturation. So by looking at HLA upregulation, which is class 1, and CD80, which is a costimulatory ligand for T cells, we indeed found that several DCRs were able to induce maturation of dendritic cells in a cell extrinsic manner, similar to LPS control matured dendritic cells in the green bars in both panels. So of course, the big question is that do these DCR T cells exhibit enhanced adoptive cell therapy efficacy? So the data that I've shown so far for the DCRs has been human data. So we generated the murine equivalents and tested them with our various assays. So here we found that our DCR T cells, our mouse DCR T cells, were also able to potently lyse tumor. And again, this is enhanced relative to the full-length control, which is just the costimulatory ligand without any modifications. So we first tested this in our AML therapy model. So for this model, we inject 4 million tumor cells into mice on day 0. By day 5, the tumor has disseminated. So we inject T cells after treating with cyclophosphamide, and then we assess for survival over a 100-day period. And again, we're creating an aggressive tumor situation for our T cells. So we're giving, in this model, only a single transfer of T cells and no supportive IL-2 therapy. So these data are ongoing, but we're happy to see these initial results that our DCRs are significantly enhancing T cell therapeutic efficacy. So here we see that none of the TCR only T cells were able, none of the mice that received TCR only T cells, which is marked as DFP, were able to survive this tumor burden. And also the full-length costimulatory ligand T cells were unable to rescue the mice, although there was a small shift in the curve with the blue line. But really the striking data are with the red, green, and orange survival curves that show increased survival from 0% to 29 to 57%. We have also initiated testing these DCRs in our pancreatic tumor model. And so we first did studies where we did takedowns and looked for our T cells in the pancreatic tumors. So if we look first in the spleen in the left panel, we see that our DCR T cells are not overly abundant within the spleen. So they're not just non-specifically proliferating and accumulating in non-tumor organs. However, in the tumor, we see a significant increase in the number of T cells relative to both TCRT only, relative to the TCRT control. So these DCRT cells are at 7 to 17-fold higher concentrations in the tumor. We've also been monitoring tumor progression. So we've been measuring subcutaneous tumors. And so we found that the tumors from our DCRT cell recipients are much smaller than those in the TCRT cell control mice. So some conclusions from our DCRs are that they provide both cell intrinsic and extrinsic co-stimulatory signals to improve T cell and endogenous immune cell anti-tumor activity. The DCR ectodomain co-stimulatory signals are delivered by tumor-targeted T cells, which themselves localize to the tumor. So we predict that the co-stimulatory signals delivered by the DCR ectodomain will be concentrated in the tumor and less likely to result in off-target effects such as autoimmunity and inflammation. And again, we have the DCR endodomain, which can provide co-stimulatory signals that are not safe to deliver systemically, such as CD28. So we've now generated over 75 intentionally engineered fusion proteins within my lab, and we are generating data sets to support clinical translation. And what we found is that these engineered co-stimulatory fusion proteins have the potential to overcome several inhibitory barriers. So we've observed enhanced metabolism of our T cells, reduced or ablated suppression by decoy mechanism with our switch receptors, reduced exhaustion of our T cells where they're able to continuously lyse tumor cells or continuously expand and proliferate in response to multiple stimulations. We've observed a lowered threshold of activation and increased intratumoral concentration. And with our new platform, with our DCRs, we're also observing cell extrinsic benefits, which we are exploring further. So, and again, we're leaving the endogenous negative receptor intact and pairing two native proteins so that we have a very low immunogenicity of this approach. So with that, I would like to thank the members of my lab, as well as our various collaborators and our sources of funding. And if you have any follow-up questions, please feel free to reach out to us. We are also looking for committed scientists to join our group. So if you're interested in that, please let me know. Thank you for your time.
Thank you, Dr. Oda, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today. I'd also like to thank our sponsor, Stem Cell Technologies, for sponsoring today's webinar. Questions submitted today and during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until October 5th, 2024. LabRoots will alert you via email when it's available for replay. And we encourage you to share that email with any one of your colleagues who may have missed today's live event. Until next time, goodbye.